I'll tell you my story about Claire. Many, many years ago, I think it was at Canberra Intensive Care Annual Scientific Meeting. It might have been Canberra, I'm not sure. A nurse won a research prize. And I went, wow. It would have been the late 90s, early 20s. But how do you get onto that? And this was Claire. So Claire is now one of the preeminent researchers on vascular access care in Australia. She established Avatar in 2007, and she's going to talk more about Avatar in just a sec. But I think we're very, very privileged to have Claire based in Australia and to know that Australian nurses, not just Australian clinicians, but Australian nurses are at the very, very forefront of developing basic research evidence for care on something that's so essential to all patients, which is vascular access care. So I'll just hand over to Claire.
Thanks, Kay. I think that's the first time I've ever had an unscripted bio, and I must say I liked that much more than the usual rubbish that people get up there and talk about, you know. Hi everyone, so great to be here. It's a nice bit colder here today than in Brisbane. It's still horrible and sweltering, but I know you've been suffering pretty badly as well this summer, hey? So yeah, it's actually our 10th birthday this year, so I've been working in research for longer than that, but we set up Avatar under its former name, which was a bit less catchy, which was the IVAD Research Group. That didn't get quite as many followers on Twitter or Facebook. But yeah, Avatar's been going for about 10 years now, so it's happy birthday to us. We always do our disclosures. Research costs a lot of money. It goes to pay our staff who do love their work, but unfortunately won't work for no money. So about a third of our money comes from NHMRC and other government grants, about a third from industry donations that are disclosed here, and about a third from sort of small hospital foundations and professional organisations. But we're not affiliated with any particular company, and we're not endorsing any particular company's products unless it's sponsored by good research, randomised trials. So vascular access, it's pretty much everywhere. Can anyone think of a clinical area within healthcare where we don't use vascular access devices? No? Silence. Yeah, I mean, sometimes people say mental health, but I mean, even in mental health, of course, people are acutely unwell, they have IVs or they have ECT and stuff. So anyone know how many IV devices are used in the world every year? Plenty? Billions, yeah. Good guess, Amanda. So we don't really know, of course, but the manufacturer, reports that are out there suggest over 2 billion devices sold every year. 2 billion. So it's a lot of product and a lot of patient experience and a lot of nursing and medical time. And of course, so many types now, so many to choose from. Every time you think you've just cracked it, they bring out a new one. And more similarities than differences, probably. I know when I was in ICU, I was very ICU focused. I only thought about central line research in ICU. I wouldn't have read an oncology CVC study. How could that relate to ICU, But I think really we put a hole in someone, we put a piece of, plastic cannula in their vein, whether they're in oncology or ICU, the catheter and the vein don't actually know which ward the patient's in. So I think there's a lot more similarity than differences between these areas. Now, I probably should go back a step. So how I got interested in vascular access really came all the way back from when I was a student. And I remember going on prac and we were allowed to watch a central line dressing. That was very exciting. I was university trained, obviously, so, to be allowed to do things. So I remember going to see the central line dressing, two nurses, full green sterile theatre gowns, masks, sterile gloves, the whole shebang. A few years later, I was working in another hospital looking after central lines. No mask, no gown, no gloves. I hope we washed our hands. But, and they thought they were doing the right thing too. So both places, different policies, same sort of line. And some people think you get into research because you don't like clinical. Well, in my case, that's not true at all. I was like a goody nurse, and I wanted to know the right way. Well, I don't care which is the right way, just tell me, I want to do it for all the patients. Similar things happened with PIVs. So remember when I was starting out, it was a 5 ml syringe, and you'd come along and give your flush every 4 hours. You had to do it every 4 hours, otherwise that PIV was going to fail. And then 5-10 years later, I'd be in a different hospital, and it would be 10 ml syringe now, you've got to use that. It's only once a day you have to flush now. And I was like, well, okay, but is that right? I don't know, which one's right? So it was all those sort of practice inconsistencies that just made me question, well, do the giving sets all need to be thrown away 3 times a week? Or is it twice a week? Or is it once a week? I want to know. And of course, it wasn't until I started, doing some postgraduate study, learning about research, learning about evidence-based practice, that I realised that these decisions shouldn't be made on, my hospital does it this way. It should be based on which one actually works, tested in a good quality trial. And of course, those trials often weren't there. So that was kind of my story of how I got into vascular access, because I was curious, really. I just wanted to know the right way to do things, the way that worked for patients. And I guess all of us that are good nurses or good doctors, that's why we go to work every day, isn't it? But, why have I stayed in research? Why has Avatar become so big? It's because of the impact. It's not just because I'm curious, because a lot of curious researchers never get any funding, never, have any success, unfortunately for them. But the reason we've had, over $10 million in research funding and keep growing and growing is because there's problems. And the problem is that of those 2 billion, remember, that we buy and use in the world, about up to 1 in 4 CVADs fail, and about 35, 45, 50%, depending on your study, of PIVs fail. So when I say fail, I just mean any complication that stops that device being able to be used for what the patient needs it for. So, of course, we think of infection a lot. That gets the most headline space. But, it's all those other really common things, like you just walk up to the PIV and it's occluded. You can't use it. Why? Who knows? It's just stopped working. Dislodgement, occlusion, thrombosis, phlebitis, pain, air embolus. I mean, the list is just goes on and on. And you can imagine, of course, the impact this has on costs. I mean, the amount of time we waste trying to troubleshoot, having to take out devices that don't work anymore, having to put a new one in, having to find someone who can put one in. So the cost is immense. And obviously for patients, I mean, interrupted therapy, delayed care, pain, worry. And of course, if they end up with, bloodstream infection and end up with multi-organ failure or something really devastating, then, that can end their life. So why is this not like, the front headline of every newspaper? Like, remember when hip replacements, there was that bad batch in the States and there was so much media about it. I mean, if the public knew that we're putting in these devices and, 1 in 2, 1 in 4 fail, I mean, that's pretty bad statistics, like why is the TGA letting us get away with this? Why is the nursing board letting us get away with this? But for some reason, vascular access has just been sort of seen as one of those things that it's so problematic that we almost think that's part of it. It just comes with using those devices. So I guess, it's not just us. Like when we first started seeing all this bad data, these high rates of failure, we thought, oh God, our hospitals are terrible. And they were. But then, we started looking further afield and realising that it wasn't just us, it was, common. So this is just some PIV studies. I just randomly went and found any study I could where they reported the failure rate of PIVs. So this 34% comes from one of our studies in Brisbane at 3 hospitals, 6,000 patients. But then I just went trolling around and I found sort of a bit lower rates in some studies in some countries. China, Lebanon, Singapore, but then I found higher rates. So Canada, Spain, Uganda, Japan in the 40%. That's getting pretty shocking. But I even found higher rates. So in the US, 53% in one study, Chile, 54%. I mean, this is more than half of their PIVs are just not lasting the distance. And I even found one very brave group of researchers from India who published their 100% failure of their PIVs in their study. So, good luck to them that they're so honest. I don't know that I would be so brave. Now, this isn't meant to say that, all of China has a failure rate of 27%. Of course it doesn't. All of these studies measured, different things. Some are reporting occlusion, some are reporting phlebitis, and definitions vary. But, you can see that consistently there is a problem. And if we pulled out the central line literature, the rates are probably sitting more around the 25% failure. But again, very consistent message coming through that there is a big problem with complications, and it's not just about infection. So that's really how we got together. So Avatar is really just a voluntary network of interested people. So people sometimes think, oh, isn't it great that Griffith is like investing in this Institute of Vascular Access and they're providing all this money? Well, there's a few of us that are paid, academic staff, but nearly everybody is either on grant money or they're paid by their hospital, paid by their university to work together. And I don't know if you can read that very well, it's a bit small, sorry, but our real goal is to make complications history. So it's audacious, of course, but you have to make these goals. So our goal is that a patient comes to hospital, they need some sort of vascular access treatment, we assess them properly, we give them the right device, they have that one device for their entire length of their therapy. It's comfortable, it doesn't have any complications, and it keeps working. So that's our goal. I think we've got a fair way before we can get there. We'll never go out of business pretty much, but we're doing our best. And you can see there's some ways to contact us or keep in touch if you like. We've got a newsletter and those sort of things. So what do we do about this failure? It's just so overwhelming and obviously has many causes, right? If it was simple, we would have cracked this a long time ago. So there's obviously heaps and heaps of practices that we do now that could help. Maybe they're making things worse. Where do we start? So we kind of think of it as this patient cycle through the vascular access journey, and there's decisions that we make as clinicians, there's interventions that we're doing that may help or may not be helping. So first we have to decide which device this patient needs. Do they need a PIV? Do they need a PIC? Which sort? Then, of course, we have to have the insertion procedure, which can trigger off a whole lot of complications both at the time and in the days down the track. Then we've got the longest period of the dwell, which actually the research often ignores, which is the functional time. So once you have it in, you might need to keep it working for weeks or months. And how do we actually make that happen? That's my son there. He's very cute, isn't he, little one? He's missing me today. Now, and then removal, of course. So even though people need these devices, they're life-saving, they're critical, obviously any device day is a risk, and we want to get them out as soon as possible, and we want to take them out in a safe manner. So this is how we structure Avatar. Avatar's quite big, and because there's so many interventions, we have these platform areas where these researchers provide sort of networking, advice, coordination. Doesn't necessarily mean they do all that research, but they're kind of the go-to person. So I'm looking after dressing and securement at the moment, which I just love. Flushing and blood sampling is Professor Samantha Keogh. She's at QUT in Brisbane, Queensland University of Technology. Marianne Wallace is up at the University of the Sunshine Coast. She looks after devices and therapy. Li Zhang is actually a medical doctor from China who then went into microbiology, and she looks after infection prevention and micro. Marie Cooke, who's here somewhere, there we go. Marie, have a wave, Marie. There she is. She's in charge of education and evidence-based practice, so this sort of thing today is under her wings. We also have some liaison people who are absolutely key to this as well. So Nicole Marsh is a very, very experienced clinical trial director and manager, and she advises a lot of people on what's feasible and what's not feasible. Gillian Ray Barul does our media and communications, so she's heavily involved in Twitter and Facebook and newsletters and so on. Trisha and Amanda, I'll get to in a minute. Amanda's been looking after the industry engagement, which is also vital. That's a massive part of our healthcare industry. And Kay is our newest appointment. Welcome, Kay. Soon to be Dr. Kay, if her thesis goes well, which I'm sure it will. Has come on board as our knowledge translator. So it's not just us, though. With us, like, we'd just be sitting at the university writing papers that, would that be of any interest? No. So really, these are the crucial people. So we reach out to, like, all of those clinicians who actually have an idea, are interested, but say, oh, yeah, I can see a study needs doing. I'd quite like to do it, but I don't know how to. I wouldn't have time to do too much, So we're really touching base with them, and then we have our academic colleagues who, the clinicians sometimes think that the lecturers and stuff would have a lot more time to research, but actually they're just hammered a lot of the time with 300 students turning up. So again, we try and reach out and connect with those people that have an interest, have skills. And somehow we bring, through the power of love or something, we bring, all of those people together and try and network and share our skills. And that's really the only way to get research done, I think. Now, newly, excitingly, we've just announced our Avatar Paediatrics platform, which we are having headed up by Amanda down the back. Wave, Amanda, Dr. Amanda, and Trisha, her buddy there. So that's a really great achievement. And actually, our paediatric colleagues often put us to shame. They do so much research, and like they're one hospital. We have about 400 adult hospitals, and they have one paediatric, and they're just putting us to shame most of the time. Okay. So the reason Avatar works, I think, is because we have people with feet in all of these camps. So I said earlier, a central line in oncology probably has a fair amount of the same complications and needs as one in ICU or the general wards and so on. So we have people really from most clinical areas, ED, oncology, ICU, etc. Then we have our non-clinical researchers, so statisticians, health economists, lab scientists, all of those people. And of course, as an academic group, we're training people all the time in their masters and their PhD. So it's quite a big group now. We do have state-based chapters, and Evan, I haven't seen Evan yet today. Where is Evan? So I'm sure a lot of you know Evan Alexandru, and he's our New South Wales chapter lead. And Kerry is here from Melbourne. Where are you, Kerry? She's up the back as well. Kerry's our Victorian chapter lead. So we're trying to sort of have someone in each state that people can go to as a first step to get advice and who's the best one to talk to. We've got about 15 PhD and honours students at the moment, looking at all sorts of wild and wonderful things, all within vascular access. And the other thing we do is advise on guidelines. So any of you who are lucky enough to be writing state guidelines or, infusing oncology nursing guidelines or ED guidelines about vascular access, you'll know how tough it is and how you're out there looking for good quality evidence so that you can put references on your guidelines. So we're often giving advice and support there. But that also means that we realise that a lot of the research out there about vascular access, a lot of the publications are not high-level evidence to really make hard decisions on. A lot of it is very much experiential, pre, post, this is how we did it, this is how we feel kind of stuff, which is fine to raise questions, but obviously not definitively answer them. So really this is our core values, I guess, or activities. We're finding problems, we're testing solutions, but we don't just want to test them, we want to actually get them into practice, and that's the sort of thing that today is about. We want to work closely with the patients and the clinicians. We're very bedside focused. The research we do, we're trying to always do the highest quality we can. We don't ignore our industry partners, recognising that they invest a huge amount in research and development of their products before they even come to market, and of course they have a great knowledge about that we can tap into. Also all the support organisations, the various colleges of nursing in the different specialties, and these days we're working very hard on knowledge tools and social media and stuff to try and help people to be able to use evidence without necessarily having to read a stack of papers, this high. Even I don't like doing that. These are a few things we're working on this year, a few new initiatives. One of them is the big conference in Perth in May. If anyone is able to get across, that'll be a fantastic few days in our second ever vascular access conference in Australia. And the knowledge translation that we have employed Kay to. So we're trying to work even harder to make sure we make our evidence accessible, useful, and keep the dialogue going with the people that are going to be using it. So that's all about intro to Avatar. Are you next, Kay, or who's? Yeah, thanks everyone. I look forward to meeting a lot of you today. Hope you have a great day.
Thanks, Claire. Oh, that's the wrong one. Okay, so this is Dr. Marie Cooke, who's the Deputy Head of School for Research at Griffith, and is looking after education and evidence-based practice for the Avatar group.
Hi everyone, it's so good to see you all here today. We flew down from Brisbane yesterday and people say, why are we doing this in Sydney? But the wonderful Kay has organised this, so I have to thank you, Kay. This has been a great achievement to get Kay on board and to start the process of trying to get the evidence from Avatar out into everyday practice, or if we don't have the evidence, talk about what we know as best practice. So thanks very much, Kay. It's been a wonderful thing. And the wonderful Kay gave me this title, Hierarchies of what? So a very mysterious title. So like any good academic, I went to Dr. Google to find out what might this mean. So this is the search that came up. Do I just do the down arrow? Where is that? This one? No? That one. So here's the search. Yeah, it did. So hierarchies of planning and hierarchies of effects. I thought, well, yeah, they're interesting, and I could see that they had some relationship to vascular access devices. Hierarchies of oppression, well, that just seemed a bit too weird and a bit too serious and too dark for what I was thinking about. I really like the sound of hierarchies of angels. That seemed to me to have some value in relation to vascular access devices. But in fact, today is about hierarchies of evidence. So I just want to spend the next little while as a refresher, really. I'm sure many of you will know this information, but it's just so that we're all on the same page when we come to talking about levels of evidence, because I know some of the presenters today will talk about when they're talking about what's available in terms of evidence, level 1, level 2 evidence. So I just really want to spend a little time refreshing on that. So this figure illustrates decisions that influence patient outcomes. So it's one that I'm sure you're familiar with. The 4 components that you see there have been identified by a number of authors, but DeCenzo and Callum, for example, have identified these 4 components as providing a simple model to guide evidence-based nursing practice. And so that combination of what is the best available evidence with your clinical expertise and the patient's values and expectations come together to help make decisions around clinical issues. But we also know that in healthcare resources are limited, and so these factors all need to be taken into consideration when we're looking at available resources. So generally, the higher the level of evidence here for best available clinical evidence, generally the stronger the evidence there is, the more comfortable you will be in using that evidence to help you with the decision that you're making about this clinical issue. So evidence comes from research studies, and the strength of the evidence, you know, comes from the number and the types of studies. I'm always going to miss this down button. I don't know why. I seem to want to go somewhere else. It must be my laptop at home. So I'm sure, and Claire raised this in her slides, I'm sure you've all seen this hierarchy of evidence with the lowest to the highest strength, with the lowest being in vitro studies and the highest being systematic reviews with a meta-analysis and level 2 randomised controlled trials. So the randomised controlled trial, level 2, or systematic reviews with a meta-analysis of randomised controlled trials is level 1 evidence. And they provide the strongest level of evidence that we have for cause and effect. So if you do this intervention, this will be the effect. And the reason why they're higher on that hierarchy is to do with hopefully their methodology and the quality of the work, so that they're controlling for bias along the way. Now, in saying that, not all systematic reviews, not all randomised controlled trials have the same quality. You do need to understand the way in which they've addressed risks of bias and things like that for you to understand the quality and therefore the validity of their results. But we don't really have time to look at grading of studies, the methodology and the risk of bias. So really today I'm just focusing on levels of evidence, not really grades within those overall levels. I suppose there is an assumption that there is strong evidence around vascular access practice. And in fact, we do know that there is really good level 1 and level 2 evidence for a lot of vascular access interventions. But we also know that there is variability in practice around those levels of practice. And we know that because of our practice audits that we've done, through anecdotal conversations that we have with clinicians. So one of the important outcomes today is that we really were wanting to start looking at how we can change that variable practice and look at trying to translate evidence into what we do. So the Avatar researchers do research within all these levels of research studies, but predominantly we do look at randomised controlled trials and systematic reviews. But as I said, now we're sort of really seeing a big role in trying to bridge that gap between what we know as strong evidence and what's happening in practice, and hence why we're here today. So thank you for that. So really here I'm just wanting to use an example from the past to illustrate the pitfalls of not looking at evidence. So this is from a publication in 1992 that looks at meta-analyses for treatments for acute myocardial infarction. So if a meta-analysis had been done, it would have made a huge difference if this was the way we had looked at the evidence. So the authors here in 1992 published a number of meta-analyses around different treatments for myocardial infarction. The one that I've used today is around thrombolytic therapy. So you'll see down this side here all the randomised controlled trials that were undertaken from the 1960s down through to the 1990s. And the next columns illustrate the cumulative number of trials and the cumulative number of patients in the trials. And then this middle section here, which many of you will know as a forest plot, although forest plots can have more information, the principles are the same, and that is this line here is the line of equal effect. And you can see from this illustration that any of the analyses on the left of that line, the treatment worked. It favoured that treatment. Any of the analyses on the right favoured the control. The dots that you see are where the main effect was, and the horizontal lines are the 95% confidence intervals. So if any of these confidence intervals cross that line of equal effect, then we can't be confident that that treatment had that effect. So that you'll see here with these analyses that these authors did, that the top 5 did cross that line. But from about the mid-1970s, all of the analyses favoured the treatment. So the treatment worked. And in fact, if we look at the meta-analysis that occurred in 1980, well, it didn't occur in 1980, it occurred in 1992, but the meta-analysis here for 22 RCTs with 5,000 odd participants showed that that treatment worked really well. It was very effective with a p-value of less than 0.001. So I suppose the really worrying thing about this is the number of studies in the blue box. So a further 41,000 patients were randomised into a trial of a treatment that was effective. And half of those, 20,000 odd, wouldn't have received that treatment. And then I suppose outside of those trials, there would have been other patients who also didn't receive that treatment. Because if you look to the left, to your right, of that forest plot, you'll see that the authors also plotted whether the evidence was in textbooks and journals and whether it was in practice. So you can see that there's a huge gap between when we knew that the treatment was effective to when it became routine practice. And that gap really hasn't lessened, and I'm sure you're all aware of that yourself. But I found a 2012 article in Archives of Disease of Childhood, and the author, Mitch Blair, estimated that the average length of time for research findings to be implemented into routine practice is 17 years. So it's still a long time. So again, the importance of today in terms of trying to get your assistance to help us begin to bridge that gap between what we know as good evidence and routine practice in vascular access care. Most of you will also know that forest plots, these usually classically use a diamond for the overarching overall effect. Thank you. So I just want to go back to this model that I started off with, and I'm sure you're aware, as do other authors identify, that your clinical expertise or the patient's values and expectations sometimes predominate in this decision process. And so, for example, your expert knowledge might indicate that the patient is too frail for a particular treatment, or there's some contraindications for a treatment, or the patient may refuse the treatment or want something else. And your expertise is based on your wonderful knowledge and your experience. But some recent work that we've been doing in Avatar would suggest that patients feel they're not being involved enough in decisions around their devices, and they're frustrated in this lack of collaborative decision making. And what I wanted to do today was just play a couple of videos, and these are two women that we interviewed on campus. One is a nurse who has a port for medical treatment, and the second person is a woman who has a port for overnight nutritional support. And their voices really give us an idea of what they expect from health professionals, but it resonates well with some survey results that we've just got in, that the qualitative comments that people put on the survey really identified their frustration with not having their voice heard about their devices. So the first one is Debbie. Just stay here.
I think it's really important that healthcare workers understand the differences between the different vascular access devices that they're working with, and in particular, what are the things around access and maintenance that are specific to particular access devices, like what's important to know when accessing a port and caring for one, and what are the specific complications that you might come against with that particular device. The other thing I think that is important from a patient perspective is healthcare workers asking patients and listening to them about what their experiences to date have been, what currently works when accessing your port, what difficulties have you experienced, and where possible incorporating and entailing that into your day-to-day management of the patient and the port.
The most important thing to me about having a vascular access device for treatment is that it is my lifeline. I infuse parenteral nutrition as a life support therapy, and it's given me an extra chance at life, for which I'm forever grateful. On the other side, however, is that it's also life-threatening with the increased risk of line sepsis. My message is to healthcare professionals about vascular access devices. In regards to insertion of vascular access devices, I very much appreciate your skill, your training, your knowledge. I too understand my body a bit, and so I would ask that you please work together with me for the best results. I appreciate explanations and information about various types of vascular access devices, the pros and cons, as well as being involved in the choice of vascular access device.
Just in summary, really, it was just looking at that evidence-based practice, a model of decision making, looking at how we can incorporate the best available evidence and the hierarchies of evidence that there is, and maybe we need to incorporate the patient more in our decisions when it comes to vascular access care. So thank you.
Okay, first lot of group work for today. So we might have to forget about the first step. So in your handout you got today, there's a page that's got the instructions for the group work on it, but I'll just go through them now. So the objective of the first lot of group work is for you to think about a recent patient who had vascular access problems, and under what headings did those problems occur? Was it the patient had poor veins? Did the clinicians choose the wrong device? Did they assign the intern to put a cannula in a difficult patient? What are the sort of things that cause the problem? So you'll be working in your tables. You need to appoint a scribe and a leader for each table, please. And just was working alone for the first 5 minutes. Could you just think about that patient? An old lady, a young lady, someone, so within your own particular clinical area, because we need to develop an understanding of the problems. So, you know, not having the emergency department just whack in any old line that's not appropriate for a patient. Then I'll get you to, within the group, go round the table talking about one barrier that you had until that list is exhausted as the scribe writes down what those problems were. You'll need that bit of paper for the second lot of group work, because then you're going to talk about what things might fix that problem or what things you've developed locally to address that particular problem. So you'll have a facilitator for your table to share. So Evan, you're facilitating tables 1 and 6. Kerry, you're facilitating tables 2 and 7. So basically behind each other. Margie, you've got 3 and 8. Amanda, you've got 4 and 9. Trish, you've got 5 and 10. Claire, you've got table 11, and Marie, you've got table 12. So just to repeat again, just quietly to yourselves think about a patient that you had a recent problem with, and then start sharing together as a group. And then at 9:45, we will then feed back to the larger group. Okay? There should be noise. Okay.